Activation energy barrier is the amount of energy needed to push the reactants. The reactants A, B, and C, D must absorb enough energy from the surroundings to reach the unstable transition state, where bonds can break. After bonds have been broken, new bonds form, releasing energy to the surroundings. How enzymes speed up reactions. Heat can increase the rate of reaction by allowing reactants to attain transition state more often, but this will not work as well in biological systems because high temperature denatures proteins and kills cells. Second, the heat would speed up all reactions, not just those that are needed. Instead of heat, organisms carry out catalysis, a process which a catalyst, for example an enzyme, selectively speeds up a reaction without itself being consumed.
One or more substrate for each type of enzyme, depending on the particular reaction. In some reactions, a single reactant substrate is broken down into multiple products. The active site is the region of an enzyme where substrate molecules bind and undergo a chemical reaction. The active site consists of residues that form temporary bonds with substrate and residues that catalyze a reaction of that substrate.
As humans, we like to keep everything under control. For example, all enzymes in our body have a temperature of 37 Celsius. Any temperature changes can cause the enzyme not to work properly. Optimal conditions favor the most active shape for the enzyme molecule. Each enzyme has an optimal temperature and an optimal pH where it functions the best. A good structure and a good function equals a good enzyme. Many enzymes are simple proteins consisting entirely of one or more amino acid chains. Others contain a non-protein component called cofactors, which are helper molecules that assist enzymes during catalysis. It is known as a non-protein chemical compound. Two types of cofactors are inorganic and organic, also known as coenzymes. Cofactors enhance enzyme activity.
We move into enzyme inhibitors, and inhibitors are what slows down or shuts down the enzyme activity. And there are two types of inhibitors, which are competitive and non-competitive inhibitors. Competitive bind to the active site of the enzyme, competing it with the substrate. So basically it gets in the way of the substrate binding, and it also looks like a substrate. Non-competitive inhibitors bind to a different site of an enzyme and resulting the changes to the enzyme shape and making active site less effective. And some examples of inhibitors are like toxins and pesticides and poisons and antibiotics. So we move into the evolution of enzymes, and since there are proteins encoded by genes in DNA, if changes or mutation occurs in genes, it leads to changes in amino acids composition of enzymes or structure. It can be both good and bad. Altered amino acids enzymes can change their substrate particularity, and this is how new enzymes evolve that catalyze different reaction with a new substrate. And under new environment condition, a novel form of enzyme might be formed or favored. And so we look over here, and there are new enzymes, and which can have a function, which can have new functions and catalyze new reactions.